Let me start. Good morning, my dear friends, my dear students. Today the topic is third and fourth ventricles of the brain. So let me start with the third ventricle of the brain. This one enlarge PowerPoint. Okay. So as you know, the ventricles are present. These are in the brain. They are also present in the heart. Okay. So these are the cavities which are present in the heart. So here also in the brain also you have got the ventricles. So here they are filled with the cerebrospinal fluid. Okay. So these ventricles are lateral ventricles in the cerebral hemispheres, third ventricles in the midbrain, and I mean between the two thalamus, and fourth ventricle between the pons, medulla, and cerebellum. So let me start with the third ventricle. So it is a midline structure. It is a midline slit-like cavity. Okay, between the thalamus and hypothalamus. So this is the cerebral hemisphere, and this is the cerebellum. Okay, this is the pons and medulla oblongata. So in the cerebral hemisphere, you have got the lateral ventricle. Okay, and here this is the third ventricle, and this one is the fourth ventricle. So today we will study about this third ventricle. Okay, it is a slit-like cavity. It is a narrow slit. Okay, unlike lateral and fourth ventricle, and it is lined by the ciliated columnar epithelium. So that is nothing but the ependyma. So the lining epithelium of the ventricles and central canal of the spinal cord is called as ependyma. So that is the epithelium is ciliated columnar epithelium. So let us study the boundaries of third ventricle. So this is the third ventricle. What are the boundaries? So it has got anterior wall, posterior wall, and lateral wall. So what is present in the anterior wall? This is the sagittal section of the brain. So anterior wall. So this is the anterior wall. This part. Okay. So anterior wall is formed by anterior column of the fornix. So this is the fornix. What is this? This is the corpus callosum. So from the corpus callosum, the fornix is passing anteriorly, and this is the anterior part of the fornix. So this forms one of the part of the anterior wall. Next, lamina terminalis. So this is the lamina terminalis. This part. Okay. So this is the lamina terminalis. Next, anterior commissure, and this is the anterior commissure. This bulging. So this part forms the anterior wall of the third ventricle. Next, posterior wall. Posterior wall is formed by pineal body. So this is the pineal body. Okay. And posterior commissure. So this is the posterior commissure. Next, cerebral aqueduct. So this is the one, cerebral aqueduct. I mean initial part of the cerebral aqueduct. See, all these three forms the posterior wall of the this third ventricle. Okay. So this is the coronal section of the brain. So in this you can see the third ventricle. So you can observe the third ventricle is like a slit, narrow slit. Okay, between the two thalamus. You will get the thalamus here, hypothalamus here. Okay. So in this diagram you can see very clearly. So this is the thalamus. Okay, right and left thalamus, and this is the hypothalamus. So this is the narrow slit like third ventricle. Okay. So coming to the lateral wall of the third ventricle. So this is the lateral wall. So what are the structures present here? Just now we saw thalamus and hypothalamus. In this diagram, in the next diagram here you can see. See, this is the lateral wall. So this is formed by thalamus. In the upper part thalamus, and in the lower part you have got the hypothalamus. That is medial surface of the thalamus. Medial surface of the thalamus. So this is medial surface of the thalamus forms the lateral boundary, lateral wall of the third ventricle. So next, hypothalamus. That is also medial surface. Okay, medial surface of the hypothalamus. Next, hypothalamic sulcus. So there is a sulcus between the thalamus and hypothalamus. So that forms the, that is also one of the part of the lateral wall. So that is between the thalamus and hypothalamus. So here you will get the hypothalamic sulcus. Okay. So next, coming to the roof of the third ventricle. So this is third ventricle and this is the roof. Okay. So this roof is formed by the ependymal lining, the undersurface of the tila choroidae. So these are the two thalamus, isn't it? So these two thalamus, it is connected by ependyma. So that forms the roof. And here you got the tila choroidae. Tila choroidae. So this ependyma lines the tila choroid of the, which is present between the two thalamus. So this projection what you are seeing is the tila choroidae. So that is lined by ependyma. Okay. So this is roof of the third ventricle. Choroid plexus projects downwards from the roof. So this is choroid plexus. Okay. And next, coming to the floor of the third ventricle. So this is third ventricle. So these are the structures which forms the floor of the third ventricle. What are the structures from anterior to posterior side? One is optic chiasma. You know the optic chiasma is formed by the crossing of the optic nerve fibers. So this is the optic chiasma in the sagittal section. Next, this is the infundibulum. Infundibulum. Next, tuber cinerarium. So here actually it's not shown here. So this is the tuber cinerarium of the, which is present in the floor. So next, mammillary bodies, posterior perforated substance and tegmentum of the midbrain. So mammillary bodies, so these are the mammillary bodies, and this is the tegmentum of the midbrain. Already we studied. This is the tegmentum of the midbrain. Next, here you have got the posterior perforated substance. Posterior perforated substance. This part. See, all these forms the floor of the third ventricle. Okay. Next, communications of the third ventricles. It communicates with the other ventricles. Okay, as you are seeing in the diagram. So the third ventricle, it communicates with the lateral ventricle. So this communication is called as interventricular foramen. Interventricular foramen of Monro. Okay, so this is interventricular foramen of Monro. There is communication between the lateral and third ventricle. And this is present between the fornix and anterior pole of the thalamus. So in between these two structures, this interventricular foramen is located. And it is also communicated inferiorly with the this ventricle. So this is the fourth ventricle. Okay. So this is third ventricle and this is fourth ventricle. Inferiorly it is communicated with the fourth ventricle. So this is called as, already we studied, cerebral aqueduct. Cerebral aqueduct or aqueduct of Sylvius. Okay, so this is the one. So this is about the third ventricles, boundaries and shape and communications. Okay, next we'll move on to the fourth ventricle. So this is the fourth ventricle. In sagittal section, what is the shape of this fourth ventricle? It is triangular in shape. So this is sagittal section. So this is triangular shaped ventricle. So this is located between the cerebellum, pons and medulla oblongata. Pons and medulla oblongata, they are present anteriorly and cerebellum is present posteriorly. Okay. So it is the cavity of the hindbrain. Okay, cavity of the hindbrain. It intervenes between the cerebellum anteriorly, pons and medulla posteriorly, and in cross section, if you remove the cerebellum in the horizontal section, it is rhomboid shape. It is rhomboid shape. So this is the one. So here the cerebellum is removed. Okay, and it has got 4 angles. What are these? Rostral angle, caudal angle, and 2 lateral angles. You can see here this is rostral, caudal, and 2 lateral angles of the fourth ventricle. It has got, what are these boundaries? 2 lateral limits, that is lateral boundaries, and roof and floor. So let us study the boundaries of floor of the fourth ventricle. So this is the floor of the fourth ventricle. Okay, what you are saying is the floor of the fourth ventricle. We'll study the boundaries of fourth ventricle. So lateral boundaries, superiorlaterally and inferiorlaterally. So superiorlaterally, what you are saying? Superior cerebellar peduncles on either side, superiorlaterally, and inferiorlaterally. These are the structures from medial to lateral side: gracile tubercle, cunate tubercle, and inferior cerebellar peduncle. So these are the 3 structures which forms the inferiorlateral boundary of fourth ventricle. So next, coming to the roof of the fourth ventricle. So in this diagram you can see, so this is the roof. This is the roof of the fourth ventricle. Okay. So it is a tent-like structure. It is a roof is tent-like structure. This is also called as posterior wall of the fourth ventricle. See, the apex of this roof projects to the cerebellar white core. See, this is the apex of the roof. This is continuous with the white matter of the cerebellum. Okay, which is projected posteriorly. It has got 2 parts, upper part and lower part. Okay, so upper part is stretched or bridged between the two superior peduncles, that is called as superior medullary velum. Okay, so lower part, it is called as inferior medullary velum. Okay, so I will show you in this diagram. So this is the upper part. Okay, so this is superior medullary velum, and this is the inferior medullary velum. So in this diagram, so this is the superior medullary velum and inferior medullary velum. Superior medullary velum, it is stretched between the superior peduncles. Okay, this is superior medullary velum, and inferior medullary velum between the inferolateral boundaries. Okay, so that superior medullary velum, it is the nervous tissue, and inferior medullary velum, it is non-nervous tissue. It is formed by pia mater with ependyma. So these 2 layers, these 2 coverings forms the inferior medullary velum. Okay. So here you have got the, in the inferior part of the roof, you have got the tela choroidae. So here you can see here. So this is the ependyma, which is the one layer which is lining inside. The outside is pyometer. So here the pyometer and ependymal lining forms the inferior medullary velum. Okay. And here in the inferior part you have got the choroid plexus of the fourth ventricle. Okay, that is called as tela choroidae. Okay, which is T-shaped. The lowest part of the roof is called as obex. So this part, the lowest part is called as obex. It covers the inferior angle of the fourth ventricle. So it will come here. This is in this can't see. There is a median aperture at the inferior angle, that is called as foramina magandi. Through this, the fourth ventricle communicates with the cerebellomedullary cister. Okay, that is called as cisterna magna. Okay, so in this you can see here there is a foramen at the inferior part of the lower part of the inferior medullary velum. Okay, so this opening is called as foramina magandi. With this, it communicates with the cisterna magna. Okay, so the CSF from the fourth ventricle is traversing to the subarachnoid space at this point. Okay, and you can observe here also at the lateral angles there are again two more apertures. These are called as lateral apertures, and the name given is foramina of Flushka. These are the foramina of Flushka. With this, the fourth ventricle communicates with the subarachnoid space at the cerebellopontine angle between the cerebellum and pons. This foramina of Flushka is present. Okay, so these are the three apertures through which fourth ventricle communicates with the subarachnoid space. And this is the tela choroidae. Okay, this is choroidal plexus, which is in the form of, it is in the T-shaped manner. Okay, which is present in the lower part of the, just below the roof of the fourth ventricle. Okay, so in this diagram we can see the foramina magandi. So this is called as foramina magandi. At the lower part in the midline you have got the opening. This is foramina magandi, and through this it communicates with the cerebellomedullary cisterna or cisterna magna. Okay. Next, coming to the floor of the fourth ventricle. So what you are seeing is the floor of the fourth ventricle. So this is also called as rhomboidus fossa because it resembles the shape of rhomboid. Floor is formed by two structures. It is formed by pons as well as medulla oblongata. That too, dorsal surface of pons and medulla oblongata. Okay. So here in the upper part, this is formed by dorsal surface of the pons, and lower part of the floor is formed by posterior surface or dorsal surface of the medulla oblongata. So in this, yeah. So this is the floor, this part. Okay, and this is the upper part and this is the lower part. See, the upper part is formed by the dorsal surface of the pons, and lower part is formed by the posterior surface of the upper part of the medulla oblongata. Okay, so this is the floor and this is the roof. So floor, you have to study, there are so many structures in the floor of the fourth ventricle. There in the midline there is a sulcus that is called as median sulcus. So this median sulcus divides the floor into two parts, that too in the upper part. First let us study the upper part. So in the midline you have got the median sulcus. Okay, so it divides into two halls, and in the at the middle horizontally you can see the some fibers. Okay, so these are called as stria medullaris. So these stria medullaris, they are white fibers. They are originating from the arcuate nucleus. Okay, they will emerge from in the near the medial sulcus and traverse laterally on either side, and they will join the inferior cerebellar peduncle. Yesterday you have studied about this one, stria medullaris, which are originating from the arcuate nucleus. Again, each half in the upper part is divided into medial eminence and lateral vestibular area. So this is median eminence, this one medial eminence, and this part is vestibular area. So in between these two there is a one sulcus is present, which is running downwards. So this is called as sulcus limitans. Sulcus limitans divides the each half into medial eminence and lateral vestibular area. Lateral vestibular area. So this is the sulcus limitans. At the upper end of the sulcus limitans, there is a triangular depression area. So this is called as superior fovea. So this is called as superior fovea at the upper end of the sulcus limitans. So this superior fovea, it touches the superior cerebellar peduncle. And after this, just below the superior cerebellar peduncle, there is small pigmented areas present. So that is called as locus ceruleus. Locus ceruleus. Okay. So medial eminence. In the medial eminence, you have got oval or round shaped structure. So that is called as facial colliculus. Facial colliculus. So this facial colliculus is because of the underlying facial nerve, which is winding around the abducent nerve. That is called as internal genu of the facial nerve. So because of those fibers, facial nerve fibers, which are winding around the abducent nerve nucleus, so this is formed. Facial colliculus is formed. Caudally, so if you trace the sulcus limitans caudally, there is one more round depression is present. So that is called as inferior fovea. That is called as inferior fovea. From the inferior fovea, the sulcus limitans runs in the medial direction, and both the sulcus limitans of the opposite side meet in the midline. Okay. So as a result, there is a formation of two triangles. One is above the sulcus limitans, that is called as hypoglossal triangle, and below the sulcus limitans, this part is called as vagal triangle. So this is hypoglossal triangle, and this part is vagal triangle. So hypoglossal triangle, so this is you got the hypoglossus nucleus is present. Below the hypoglossal triangle, you got the hypoglossal nucleus, and below the inferior fovea, that is you have got the vagal trigone or vagal triangle. So here you have got the nucleus of vagus nerve, ninth nerve nucleus, and 11th cranial nerve nucleus. So these three nuclei are present here. So here hypoglossus nucleus is present, and here you got the vagus nerve nuclei, glossopharyngeal nerve nuclei, and cranial part of the accessory nerve nucleus. So in this vagal triangle. Okay. So again, this in the vagal triangle, if you observe carefully, you got one line. So that is called as funiculus separans. Funiculus separans. Okay. So this divides the vagal triangle into upper and lower part, and the lower part is called as area of postrema. So this part is called as area of postrema. So this area of postrema is present immediately above the gracile tubercle. Above the gracile tubercle, it is related to the gracile tubercle, area of the postrema. So in the area of postrema, you got the omitting center, okay, which is responsible for omitting. So this is the actual picture of the dissected part of the floor of the fourth ventricle. Okay. And here at the inferior angle, already you studied, there is a covering that is called as obex. That is called as obex, which covers the inferior angle of the fourth ventricle. And on the inner side of the infralateral boundary, you have got a white fibers. So these white fibers are called as tinea, and these tinea of the both sides, they will fuse at the over the inferior angle, and these white fibers, they form the obex, which covers the inferior angle of the fourth ventricle. Okay. Next, coming to the applied aspect of the fourth ventricle. So in the fourth ventricle, you will get the tumors. So these tumors are due to the ependymal lining cells, and that tumor is called as ependymoma. Ependymoma. And as a result of this ependymoma, that is the space occupying lesion. So as a result, it will obstruct the flow of the cerebrospinal fluid. So when the CSF flow is obstructed, there is lot of CSF is collected within the ventricles of the brain. So as a result, brain will enlarge. So that is called as hydrocephalus. So the baby will look like this, big head, enlarged head. Okay. So this is the normal CT, and this one you can see here, the lateral ventricles are enlarged. Okay, because of the excessive collection of the cerebrospinal fluid. So here you can see this is the normal ventricles, brain with normal ventricles, and this is the brain with enlarged ventricles. Okay. So that condition is called as hydrocephalus. So what is the treatment? They have to remove this one CSF, excessive CSF. So for that they will do the ventriculoperitoneal shunt. So through that shunt, the excessive CSF is removed, and it will go to the peritoneal cavity of in the abdomen. So that is VP shunt. So next you got the medulloblastoma. So this medulloblastoma is highly malignant. Okay. So this medulloblastoma arise from the primitive neuroectodermal cells. So if the primitive neuroectodermal cells proliferate, that will cause the medulloblastoma. So this will cause the pressure symptoms over the cerebellum. So as a result, you will get so many signs and symptoms. That is cerebellar. That is called as cerebellar syndrome. So this is about the medulloblastoma. Next, coming to the questions of this today's topic, you will get the short essays. Describe the third ventricle, and second one is floor of the fourth ventricle. Third one is rhomboid fossa. And next, short notes, rhomboid fossa boundaries. You have to mention only boundaries of the rhomboid fossa. That is 3 marks. Second question is name the structures in the floor of the fourth ventricle. Already we studied. And roof of the fourth ventricle. Question is mention foramina in the fourth ventricle. And fifth one is facial colliculus. So these are the questions which you will get in the from this topic. So thank you one and all. I hope everybody is here in the class. So let me repeat this floor of the fourth ventricle. My God. So, floor is formed by the dorsal surface of the pons and medulla oblongata. So in this diagram you can see very, so this is the floor, okay. So it is formed by the dorsal part of the pons, upper part, and lower part is formed by the dorsal part of the medulla oblongata, okay. So in the midline you have got the median sulcus, okay, from the superior angle to the inferior angle. This median sulcus is extending, the dotted line. So it divides the floor of the triangle, floor of the fourth ventricle into two equal halves, okay. And between the two lateral angles, horizontally there is passage of white fibers. So this is called as stria medullaris. So this is the one. So these are nothing but the fibers emerging from the arcuate nucleus, okay, and they will join the laterally, they will join the inferior cerebellar peduncle. And in the upper part of the floor of the fourth ventricle, you will get one sulcus limitans. So that is also coming in the lower part also. So this is called as sulcus limitans. So this sulcus limitans in the upper part of the floor of the fourth ventricle divides into medial eminence and laterally vestibular area, okay. So medial eminence, it has got a circular or oval shaped structure that is called as facial colliculus. This is because of the internal genu of the facial nerve, that is facial nerve fibers, they will wind around the abducent nucleus. So because of that, those structures, the facial colliculus is formed. Sulcus limitans at the upper end, it has got a triangular depressed area that is called as superior fovea. And the sulcus limitans will continue as locus ceruleus at the inner part of the superior cerebellar peduncles. And in the lower part of the floor, the sulcus limitans, it has got a circular depression area that is called as inferior fovea. From the inferior fovea, the sulcus limitans, it runs in the medial direction, and the sulcus limitans of both sides, they will meet in the midline. As a result, two areas are formed. Above the sulcus limitans, this is called as hypoglossal triangle, and below this, it is called as vagal triangle. So this is hypoglossal triangle, and this is the vagal triangle. So hypoglossal triangle, below that you have got the hypoglossus nucleus, and in the vagal triangle you have got the 10th, that is vagus nerve nucleus, glossopharyngeal nerve nucleus, and cranial part of the accessory nerve nucleus. So again vagal triangle, there is a line that is called as funiculus separatus. So below the funiculus separatus, this area is called as area postrema. So here you got the vomiting center. And on the inner aspect of the inferolateral boundary, there are white fibers. These are called as tinea, and these tinea will meet at the inferior angle, and at this inferior angle, this tinea will cover the inferior angle. This part is called as obex. Okay, so this is about the floor of the fourth ventricle, which is very important. Okay, with this I'll finish today's class. Thank you one and all. ಸರ್ ನೋಡ್ತಿರ್ತಾರ ಏನು ಮಾಡ್ಲಿಕ್ಕೆ ನೀವು
Very good morning to all. Today I am doing mechanism of urine formation and glomerular filtration in the significance of a renal clearance. Coming to the formation of a urine. The steps involved in the formation of the urine are glomerular filtration, tubular reabsorption and tubular secretion. These are the three steps for the formation of the urine. In glomerular filtration, the plasma which is filtered in the glomeruli and the substances reach the renal tubules along with the water as filtrate. Tubular reabsorption is 99% of filtrate is reabsorbed in different segments of the renal tubules. Next, tubular secretion. Some substances are transported from the blood into the renal tubule. With all these changes, the filtrate will become a urine. Now, we will see what is glomerular filtration. Urine formation is a blood cleansing function. Normally, the 1300 ml of a blood, that is 26% of a cardiac output, which enters a kidney for a filtration. Kidneys will excrete the unwanted substances along with the water from the blood as a urine. The normal urine output is 1 to 1.5 liter per day. Now, we will see the process, how the urine is formed. When the blood passes through the glomerular capillaries, the plasma is filtered into the Bowman's capsule, which is called as a glomerular filtration. This filtrate from the Bowman's capsule passes through the tubular portion of the nephron. Many wanted substances like glucose, amino acid, water, electrolytes are reabsorbed from the tubules is the second step, that is tubular reabsorption. Here it is glomerular filtration is the first step, whereas tubular reabsorption is the second step of a urine formation. Third is some unwanted substances are secreted into the tubule from the peritubular blood vessels. The process called as tubular secretion or tubular excretion. This is the third step. This covers the three steps of a urine formation. One is glomerular filtration, second is reabsorption, third is tubular secretion. This is a diagram showing a glomerular filtration from the afferent and efferent arterioles into the Bowman's capsule. From the tubule, it is a reabsorption into the capillaries. Third is tubular secretion, that is from capillary to the tubule, the unwanted substances are secreted into the tubule. Coming to glomerular filtration, it is a process by which blood is filtered while passing through the glomerular capillaries by filtration membrane. Now, we will see what is this filtration membrane. This is the first process of a urine formation. In filtration membrane, it is formed by the three layers of a membranes. One is glomerular capillary membrane, second is basement membrane, third is visceral layer of a Bowman's capsule. The pressures which determine this filtration fraction are glomerular capillary pressure, second is colloidal osmotic pressure in the glomeruli, third is hydrostatic pressure in the Bowman's capsule. These pressures which determine GF are either by favoring or opposing the filtration. These are, as you know, these are all the Starling forces. Next is GFR. This can be asked for a 3 marks or a 5 mark question in your exams. GFR is defined as a total quantity of a filtrate formed in all the nephrons of both the kidneys in the given unit time. I repeat, glomerular filtration rate is defined as total quantity of a filtrate formed in all the nephrons of both the kidneys in the given unit time. Normally, it is 125 ml per minute or 180 liters per day. Next, filtration membrane. As I told in the previous slides, it is formed by the three membranes. One is glomerular capillary membrane, second is basement membrane, third is visceral layer of a Bowman's capsule. Glomerular capillary membrane is formed by the single layer of endothelial cells which are attached to the basement membrane. The capillary membrane has many pores which is called as filtration pores with a diameter of 0.1 microns. Second is basement membrane. The basement membrane of a glomerular capillaries and the basement membrane of a visceral layer of a Bowman's capsule fused together. This fused basement membrane separates the endothelium of the glomerular capillary and the epithelium of the visceral layer of a Bowman's capsule. where the pedicles will interdigit in leaving the cleft-like spaces called as a slit pores, also called as a filtration slit. This filtration will take place through these slit pores. This is a diagram showing a slit pores. This is a basement membrane. These are all the podocyte cells with the slit pores in between where the filtration will take place. Third is a visceral layer of a Bowman's capsule. This layer is formed by a single layer of a flattened epithelial cells resting on the basement membrane. Each cell is connected with the basement membrane by cytoplasmic extension called as pedicles or feet. Epithelial cells with the pedicles are called as a podocytes. Coming to the process of formation of a glomerular filtration. When the blood is passes through the glomerular capillaries, the plasma is filtered into the Bowman's capsule. All these substances of plasma are filtered except the plasma proteins. So, it is called as a glomerular filtration. It is also called as ultrafiltration because all the minute particles are filtered, but the plasma proteins, which are the larger molecular size, won't be crossing the filtration membrane. Now, we will see the method of collection of this glomerular filtrate. This glomerular filtrate is collected in experimental animals by micropuncture technique by using a insertion of a micropipette into the Bowman's capsule and aspirating a filtrate. This is a filtration fraction. Filtration fraction is a ratio between the GFR, that is glomerular filtration rate, to the renal plasma flow, which is expressed in the percentage because of which we are multiplying with 100. So, 125 is GFR, 650 ml per minute is a renal plasma flow, which gives in percentage 19.2%. Normally, this filtration fraction varies from 15% to 20%. Now, we will see the pressures which affect this filtration. The pressure gradient which are responsible for a filtration, the forces like Starling forces, as I told, is a glomerular capillary hydrostatic pressure, that is blood pressure inside capillary tends to move fluid out of the capillary into the Bowman's capsule. I repeat, blood pressure inside the capillary tends to move the fluid out of capillary into the Bowman's capsule is glomerular capillary hydrostatic pressure. Second one is capsular hydrostatic pressure. It is the pressure of a filtrate already in the lumen is capsular hydrostatic pressure. Third one is glomerular capillary osmotic pressure, also called as Bowman's capsule osmotic pressure, that is BCOP. GCOP or BCOP is the same. Osmotic pressure caused by proteins in the blood favors a fluid movement into the capillary from the lumen. This is greater at the end of the glomerular capillary than at the beginning because fluid leaving the capillary will enter the lumen. This filtration fraction is 10 mmHg. This 10 mmHg we got by GCHP, that is glomerular capillary hydrostatic pressure is around 50 mmHg, minusing with capsular hydrostatic pressure around 10 mmHg. 50 minus 10 is 40, minusing with Bowman's capsule osmotic pressure is 30 mmHg, which gives a 10 mmHg of a filtration pressure. So, this is a filtration pressure of a 10 mmHg. Coming to the factors regulating or affecting this GFR. Renal blood flow. Renal blood flow, in the previous class we have know that there are two mechanisms. One is myogenic mechanism, second one is tubuloglomerular feedback mechanism. These factors also affect the GFR. Tubuloglomerular feedback ಬರ್ತಿದೆ ಮ್ಯಾಮ್. Filtration pressure, the pressure gradient responsible for a filtration forces are glomerular capillary hydrostatic pressure, that is the blood pressure inside the capillary tends to move fluid out of the capillary into the Bowman's capsule. It's around 50 mmHg. Then capsular hydrostatic pressure, pressure of a filtration fraction, okay ma'am. Filtration fraction of a renal plasma which becomes a filtrate, it's a ratio of GFR to the renal plasma flow. It's expressed in the percentage because of which we are going to multiply by 100. GFR is 125 ml per minute. Renal plasma flow is 650 ml per minute, which gives a 19.2%. So normally filtration fraction varies from 15 to 20%. Coming to filtration pressure. The pressures like glomerular capillary hydrostatic pressure, the blood pressure inside the capillary tends to move fluid out of the capillary into the Bowman's capsule, which is around 50 mmHg. Second is capsular hydrostatic pressure, the pressure of filtrate already in the lumen. It is around 10 mmHg. Third is glomerular capillary osmotic pressure. It's 30 mmHg, which is also called as a Bowman's capsule osmotic pressure, which is greater at the end of the capillary than at the beginning because of a fluid leveling capillary entering into the lumen. I repeat, it's a glomerular capillary hydrostatic pressure, that is blood pressure inside the capillary tends to move fluid out of the capillary into the Bowman's capsule around 50 mmHg. Then capsular hydrostatic pressure, its pressure of filtrate already in the lumen, it's around 10 mmHg. Third is glomerular capillary osmotic pressure, also called as Bowman's capsule osmotic pressure, which is greater at the end of the glomerular capillary than at the beginning because of a fluid leveling capillary entering the lumen around 30 mmHg. All this we have got a filtration of 10 mmHg by minusing 50 minus 40 is 10, that is filtration pressure. Coming to factors regulating a GFR. First is renal blood flow, that we have learnt in the last class, like myogenic mechanism and tubuloglomerular feedback, which is an auto-regulation mechanism for a renal circulation. This is a flowchart showing a tubuloglomerular feedback where the increase in GFR, which causes increase in the sodium chloride concentration in the renal tubule, which are sensed by macula densa, which is a part of cells of a JG apparatus, that is juxtaglomerular apparatus, which releases adenosine. This adenosine causes a constriction of a afferent arteriole, which in turn decreases the glomerular blood flow. Is by tubular mechanism, feedback mechanism, it decreases the GFR. This is how this mechanism will decrease the increase GFR. Third one is glomerular capillary pressure. Fourth, colloidal osmotic pressure. Fifth one is the hydrostatic pressure in Bowman's capsule. Sixth, constriction of a afferent arteriole. Seventh, constriction of a efferent arteriole. Eighth one, systemic arterial pressure. Ninth is sympathetic stimulation. 10th is surface area of capillary membrane. 11 is permeability of capillary membrane. 12th, contraction of glomerular mesangial cells. 13th is hormones are the other factors. I repeat the factors regulating a GFR. One is renal blood flow, second is tubuloglomerular feedback mechanism, third glomerular capillary pressure, fourth colloidal osmotic pressure, fifth hydrostatic pressure in Bowman's capsule, sixth is constriction of afferent arteriole, seventh is constriction of efferent arteriole, eighth is systemic arterial pressure, ninth is sympathetic stimulation, 10th is surface area of capillary membrane, 11th is permeability of capillary membrane, contraction of glomerular mesangial cells, 13th is hormonal or the other factors. Then if GFR is too high, it is understood that needed substances cannot be reabsorbed quickly enough or which are more lost in the urine. If GFR is too low, everything is reabsorbed including a waste that are normally disposed of is also observed. Coming to tubular reabsorption. This is the second step of a urine formation. Till now you have read about GFR, glomerular filtration rate, factors regulating a glomerular filtration and filtration fraction. Then tubular reabsorption. It is a process by which water and other substances are transported from renal tubules back to the blood. When the glomerular filtrate flows through the tubular portion of a nephron, both the quantitative and qualitative changes takes place. Large quantity of water, more than the 99% of water and electrolytes, other substances are reabsorbed by the tubular epithelial cells. Coming to tubular secretion. This is the third step of a urine formation. Tubular secretion is a process by which a substance are transported from blood into the renal tubules, also called as tubular excretion. It is from secretion from capillaries to the nephron. For excretion is tubular secretion. Here are some examples of tubular secretion. The potassium ion is secreted actively by sodium potassium pump in proximal and distal convoluted tubule and collecting ducts. Second is ammonia is secreted in the proximal convoluted tubule. Hydrogen ions are secreted in the proximal and distal convoluted tubules. Maximum hydrogen ion secretion occurs in the proximal tubule. Fourth is urea, which is secreted in the loop of Henle. Thank you. the class. So, students are to repeat. Steps involved in the urine formation are glomerular filtration, second is tubular reabsorption, third is tubular secretion. First, this glomerular filtration is plasma is filtered in the glomeruli and the substances which reaches the renal tubule along with the water as a filtrate. Second is tubular reabsorption. It's 99% of a filtrate is reabsorbed in different segments of a renal tubules. Secretion is some substances which are unwanted transports from the blood into the renal tubule. With all these changes, the filtrate will become a urine. Coming to glomerular filtration, the urine formation is a blood cleansing function. Normally about 1300 ml of blood, that is 26% of a cardiac output enters the kidneys. Kidneys will excrete the unwanted substances along with the water from the blood as a urine. The normal urine output is 1 liter or 1 to 1.5 liter per day. Coming to the process of formation, when the blood passes through the glomerular capillaries, the plasma in the filtered into the Bowman's capsule, the process called as glomerular filtration. The filtrate from the Bowman's capsule passes through the tubular portion of the nephron. Many wanted substances like glucose, amino acids, water and electrolytes are reabsorbed from the tubules. This process is called tubular reabsorption. It is the second step of a urine formation. Third is some unwanted substances like which are secreted from capillary into the tubule for excretion is tubular secretion or excretion. These are three steps like glomerular filtration, tubular reabsorption and tubular secretion. This is a diagram showing a filtration. There is glomerular filtration from afferent and efferent arterioles. This is a Bowman's capsule where the reabsorption occurs from the tubule to the capillaries and secretions from capillaries to the tubule for excretion of the unwanted substances. Then glomerular filtration is the process by which blood is filtered while passing through the glomerular capillaries by filtration membrane. It is the first process for a urine formation. For this filtration, we require a filtration membrane. It is formed by three layers: glomerular capillary membrane, second is basement membrane, third is visceral layer of a Bowman's capsule. The pressures which determine this filtration are glomerular capillary pressure, second is colloidal osmotic pressure in the glomeruli, third is hydrostatic pressure in the Bowman's capsule. These pressures which determined by favoring or opposing this filtration. Now we will see what is GFR. It is defined as a total quantity of a filtrate formed in all the nephrons of both the kidneys in the given unit time. Normally it is 125 ml per minute or 180 liters per day. I repeat the definition. It is defined as total quantity of filtrate formed in all the nephrons of both the kidneys in the given time. Normally is 125 ml per minute or about 180 liters per day. Then filtration membrane, as I told before, it is a glomerular capillary membrane, basement membrane, visceral layer of Bowman's capsule. Glomerular capillary membrane is formed by a single layer of endothelial cells which are attached to the basement membrane. The capillary membrane has many pores which are called as filtration pores with a diameter of 0.1 microns. Second is basement membrane. The basement membrane of glomerular capillaries and the basement membrane of visceral layer of Bowman's capsule fuse together. This fused basement membrane separates the endothelium of glomerular capillaries and the epithelium of the visceral layer of a Bowman's capsule. In between that, the pedicles will interdigit, leaving a small cleft called as filtration slit or slit pores through which a filtration will take place. This is a diagram showing a basement membrane with porous sides. In between, slit pores are there where the filtration will takes place. Third is visceral layer of a Bowman's capsule. This layer is formed by a single layer of a flattened epithelial cells resting on a basement membrane. Each cell is connected with a basement membrane by cytoplasmic extension called as pedicles or feet. Epithelial cells with the pedicles are called podocytes. The process of glomerular filtration. When the blood passes through the glomerular capillaries, the plasma is filtered into the Bowman's capsule. All the substance of plasma are filtered except the plasma proteins. The filter fluid is called as glomerular filtrate. Ultrafiltration. Glomerular filtration is also called as ultrafiltration because even a minute particles are filtered, but the plasma proteins, because of their larger molecular size, they are not filtered. Method of collection of this glomerular filtrate are micropuncture technique. This technique involves the insertion of micropipette into the Bowman's capsule and aspiration of a filtrate. This is a method for a collection of a glomerular filtrate. Coming to filtration fraction. It is a ratio between GFR to the renal plasma flow into 100. GFR is glomerular filtration rate. It's around 125 ml per minute to 650 ml per minute is the renal plasma flow multiplied by 100, which gives 19.2%. Normally, this filtration fraction varies from 15% to 20%. Coming to filtration pressures. These are the three pressures which measures this filtration pressure. First one is glomerular capillary hydrostatic pressure. The blood pressure inside the capillary tends to move fluid out of the capillary into the Bowman's capsule. This pressure is around 50 mmHg. Second is capsular hydrostatic pressure. The pressure of filtrate already in the lumen, that is around 10 mmHg. Third is glomerular capillary osmotic pressure. That osmotic pressure caused by proteins in the blood favors a fluid movement into the capillary from the lumen, also called Bowman's capsule osmotic pressure, which is greater at the end of glomerular capillary than at the beginning because the fluid leaving the capillary and entering the lumen, that is around 30 mmHg. So, filtration pressure is calculated by GCHP, that is glomerular capillary hydrostatic pressure minus capsular hydrostatic pressure minus glomerular capillary osmotic pressure. We get 10 mmHg of a filtration pressure. Next is factors regulating a GFR. First is renal blood flow. This renal blood flow is auto-regulation by two mechanisms, that is myogenic mechanism and tubuloglomerular feedback mechanism. This is a flow chart showing a tubuloglomerular feedback mechanism where the increase in GFR will increases the sodium chloride concentration in the renal tubule, which are sensed by the macula densa. These are the cells of a JG apparatus which releases adenosine. This adenosine in turn causes a constriction of afferent arteriole, decrease in glomerular blood flow is tubuloglomerular feedback mechanism to decrease the GFR. Third is glomerular capillary pressure. Fourth, colloidal osmotic pressure. Fifth is hydrostatic pressure in Bowman's capsule. Constriction of afferent arteriole, constriction of efferent arteriole, systemic arterial pressure, sympathetic stimulation, surface area of the capillary membrane. 11th is permeability of capillary membrane. 12th, contraction of a glomerular mesangial cells. 13th is hormonal factors which affect this GFR. Coming to this, if the GFR is too high, that means the needed substances cannot be reabsorbed quickly enough or lost more in the urine. The wanted and unwanted, everything is lost in the urine when there is a GFR is too high. GFR is too low means everything is reabsorbed, even unwanted and wanted substances which are supposed to be disposed are also reabsorbed when there is GFR is too low. Coming to tubular reabsorption. It is a process of reabsorption of water and other substances are transported from the renal tubules back into the blood. When the glomerular filtrate flows through the tubular portion of a nephron, both the quantitative and the qualitative changes will occur. Large quantity of water are reabsorbed, that is 99% of water is reabsorbed along with the electrolytes and other substances reabsorbed in the tubular epithelial cells. Next, tubular secretion is the third step for a urine formation, where the substances, unwanted substances are secreted from the capillaries to the nephrons, tubules for excretion. Here are some examples for a secretion. Potassium, which is secreted by sodium potassium pump in PCT and DCT, that is proximal and distal convoluted tubules, with along with collecting ducts which secretes into the lumen for a excretion. Second is ammonia secreted from the proximal convoluted tubule. Third is hydrogen ion secreted in the proximal and distal convoluted tubule. Maximum hydrogen ion occurs in the proximal tubule. The urea, which is secreted in the loop of Henle. So these are steps for a urine formation. Here mainly I concentrated on GFR. So thank you. ಮಾಡೋಣ ಹೋಗೋಣ. ಹಾಂ. ಇಲ್ಲಿ ಬಿಡಿ. ಆಮೇಲೆ ಮಾಡೋಣ. Good afternoon, students. Today we are starting with a new topic, which is nucleotide metabolism, right? As you already know, nucleotide is made up of nitrogenous base, a pentose sugar, and a phosphate. I hope there is no confusion. Whereas nucleoside, sorry, there's a mistake here. Nucleoside is made up of nitrogenous base and pentose sugar without any phosphate group. So, purines and pyrimidines are the nitrogenous bases which are present in nucleic acids. These are nutritionally non-essential components, which we mean to say that they are endogenously produced using the raw materials and degraded. Okay. So, dietary purines and pyrimidines are neither converted to nucleotides nor incorporated into nucleic acids. And while speaking about the degradation, the purine bases are oxidized to uric acid, while the pyrimidine bases are oxidized to carbon dioxide and ammonia, which are then subsequently excreted in urine, which is a major route, and a little part of it is also excreted in stools. Now, the pentose sugar has been dealt in detail during carbohydrate metabolism classes. Phosphate has been discussed in mineral metabolism, and now it's high time that we discuss about the nitrogenous bases, how they are metabolized inside the body. And as you know, metabolism includes anabolism and catabolism. So we are right now focusing on the anabolism part of it, that is biosynthesis of purine nucleotides. The nitrogenous bases, as you know, are of two types, purines and pyrimidines. Purines are adenine and guanine. Pyrimidines are cytosine, thymine, and uracil. So we right now concentrate on how exactly are these purines, that is adenine and guanine, are synthesized using the raw materials inside the body. So this is the pictorial representation of the purine ring, which is a 9-membered 2-ring structure. So we need to recapitulate a little about nucleotide chemistry before we move on to the metabolism proper. So when we speak about the bases getting attached in a nucleotide, the bases are attached to corresponding pentose sugar by N-glycosidic bond. Okay, the C1 of pentose sugar, that is the ribose sugar, okay, or the deoxyribose sugar, okay, will get attached to N9 of purine or N1 of pyrimidine. So you should remember that the numbering of the purine ring starts in an anti-clockwise manner, starting at the first nitrogen. Okay. Then once it finishes the first benzene ring, then you have 7, 8, and 9 in the second ring. Okay. So nucleoside is basically made up of a nitrogenous base and a sugar. So accordingly, if the base is adenine, we call that nucleoside as adenosine. Similarly, guanosine, uridine, cytidine, inosine, and xanthosine. Okay.
So how do we get nucleotide? By adding a phosphate group to the already existing nucleoside, which is nothing but a combination of nitrogenous base and a sugar. So adenosine, when combines with phosphate, forms adenosine monophosphate. It can also form diphosphate and triphosphate forms depending upon the needs of the body. So the simplest nucleotides are the monophosphates of various nucleosides. So AMP, adenosine monophosphate, GMP, CMP, UMP and TMP are the simplest nucleotides in nature. Okay. So these are the ring structures of purine and pyrimidine rings. Purine ring, as I already told you, is a 9-membered ring structure, and the numbering starts in anti-clockwise manner. And the sugar units present in nucleotides can be of ribose or deoxyribose. If it is RNA, it is ribose. If it is DNA, it contains deoxyribose. And you have to remember that the numbering of the carbon atoms in sugar, that is the pentose sugar, is primed. Okay. So when we are referring to prime numbers, you should remember that it's with respect to the pentose sugar. Now, coming into the details of purines, this is the ring structure of general ring structure of a purine ring, and here is adenine and guanine. Now, before we move on into the actual process of biosynthesis of purine nucleotides, we need to know the contributions of different atoms in a purine ring structure. Nitrogen at the first position, that is N1, is contributed from aspartate. So you can remember that A, that is the first alphabet of the letter, contributes the first atom. So N1 is contributed by aspartate, amine group of aspartate. Okay, and the C2 and C8, that is carbon 2 and carbon 8, are donated by N-formyl tetrahydrofolate. N3 and N9, okay, are contributed by the amine group of glutamine. Okay, so the two nitrogens at the third and the ninth position are contributed by the glutamine. Then C4, C5 and N7 are contributed by the glycine, which is the simplest amino acid, and C6 is contributed merely by carbon dioxide. So I repeat, so N1 of purine is contributed by the amino group of aspartate. C8 and C2 are contributed by the N-formyl tetrahydrofolate. The nitrogens at position 3 and 9 are contributed by glutamine. Okay, these two are amine group containing amino acids. C4, C5 and N7 are contributed by glycine, and C6 is solely contributed by carbon dioxide. Now, how do we synthesize the nucleotide? Okay. So the two purine nucleotides which exist in nucleic acids are adenosine monophosphate and guanosine monophosphate. You should remember that the nucleotide exists as monophosphate form in nucleic acids. Okay, if only when they have functions other than the formation of nucleic acid that they exist as diphosphates and triphosphates. Excuse me.